Hello viewers, welcome to my channel Biology Notes. Today I will discuss about different damaged DNA repairing systems with special reference to direct reversal repair system. DNA can be damaged from sources both inside the cell and outside. This category may be replication, reactive oxygen and so forth, and this category may be environmental like ultraviolet light, ionizing radiation, mutagens, etc. So, you might be wondering how life has managed to survive and thrive for billions of years. The fact is that organisms ranging from bacteria to humans to plants can efficiently repair their DNA. Here I have shown different DNA damage repair mechanisms. Direct reversal repair system. Under this, light dependent repair or photoreactivation, repair of alkylation damage. Single strand damage repair system. Under this, mismatch repair by DNA polymerase proofreading, excision repair of DNA damage. It is of two types, base excision repair and nucleotide excision repair. Repair of double strand breaks. Under this, non-homologous end joining, microhomology mediated end joining, homologous recombination. Post-replication repair. Under this, methyl-directed mismatch repair, error-prone repair. Under this, translation DNA synthesis and SOS response. I will try to explain all the mechanisms in upcoming videos. Today I will discuss about light-dependent repair or photoreactivation process. Direct reversal repair system. The most straightforward way to repair a lesion is to reverse it directly, thereby regenerating the normal base. Although most types of damage are essentially irreversible, lesions can be repaired by direct reversal in a few cases. Light dependent repair or photoreactivation, repair of alkylation damage. So today I will discuss light dependent repair or photoreactivation process. Here in this diagram I have shown light dependent repair of damaged DNA. It is also known as photoreactivation. This repair mechanism is seen in bacteria only, such as E. coli. Ultraviolet light induced thymine or other pyrimidine dimers are reverted directly to the original form by an enzyme called photolyase in presence of visible light. Now, see this diagram. Suppose this is a double-stranded DNA. When DNA is exposed to ultraviolet light, thymine dimers are produced by covalent cross-linkages between adjacent thymine residues. Then, DNA photolyase enzyme encoded by PHR gene recognizes and binds to thymine dimers in DNA. This binding occurs in the dark, but it cannot catalyze cleavage of the bonds joining the thymine moieties. To cleave these thymine dimers, it requires visible light, specifically light within the blue region of the spectrum. Then only the photolyase is activated by taking photon of light and splits the dimers apart. Photolyase also splits cytosine dimers and cytosine thymine dimers. Thus, when ultraviolet light is used to induce mutations in bacteria, the irradiated cells are grown in the dark for a few generations to maximize the mutation frequency. Bacterial strains with mutations in the PHR gene are defective in light repair. Photolysis has been found in bacteria and in simple eukaryotes but not in humans. So, this is all about the light dependent repair of damaged DNA or photoreactivation process. If you have any query, please comment, share the video if you like, subscribe the channel for more new videos. Stay with me, keep watching. Thank you.